First, I just want to thank Andrea, Steve, and Eric for inviting me to speak here, and I'm excited to give my talk on protease inhibitors that prevent protease-mediated cytotoxicity. And again, I'm an MD PhD student at Columbia in Alex Chavez's lab. All right, so let's get started. So just thinking about therapeutics for SARS-CoV-2 and beyond, kind of what we were thinking about when we started this project is we need effective therapeutic strategies to prohibit disease progression in COVID-19. We also need this in addition to testing, tracing, and vaccines. But taking a step back, SARS-CoV-2 is just one of 3 deadly coronaviruses to spread among humans in the last 2 decades. And there's hundreds of other coronaviruses that circulate in other species, and many with the potential to transmit to humans. And so while we need therapeutics now, we also need to be proactive against potential future outbreaks. And so we started this project with the question of how can we identify broadly active therapeutics that might be useful in the future. And so when we started developing our therapeutic search, we wanted to focus in on a target, and we focused in on the 3CL protease that's present in coronaviruses. And from proteases that cleave the polyprotein, it cleaves the polyprotein sites, while the PLP, the other protease, cleaves at 2 or 3 sites. And the 3CL protease actually exists as a dimer and actually has this nice heart shape. And functioning of the 3CL pro is essential for viral replication, which makes it a great target. And not just us is interested in this, a number of other groups are interested in it. And actually Pfizer has started a phase 1 trial for their SARS-CoV-2 3CL pro inhibitor. And a step back and looking at the utility of protease inhibitors, they've been useful in HIV and hepatitis C. So just taking a look at current methods to identify 3CL pro inhibitors, this is non-comprehensive, but probably the main workhorse is using a purified protease in vitro assay, where you take a protease, you cleave a substrate, and that releases a fluorophore, which you can detect. And this is a nice, scalable, and robust assay that's compatible with high throughput screening. But there are some downsides to this assay. One, purification is not trivial, and each individual protease, if you want to test multiple, may need unique conditions. Second, compounds are tested in a non-physiological environment. And then when you do move them to a physiological environment, they may have specificity for other targets, which may preclude your conclusions. By testing compounds against a live virus, and this is the gold standard. Does the compound inhibit the growth of the virus, which is really what we're all interested in? So it's the most meaningful physiologically. It does carry a risk of a potentially deadly virus, and also in previous talk require specialized equipment. Not virus strains are accessible or they have significant barriers, both institutionally and all defined. Infection of cells with virus doesn't always lead to a clear phenotype, and you may need to modify the virus. And finally, compounds can work by multiple mechanisms. So you could have compounds that inhibit the host anticipated this through instances. So you can't necessarily deduce what exactly the compound is doing. So our lab is mainly a technology development lab. And so we think a lot about assay development, and we try to approach problems with specific goals. So one, we want to use widely accessible and inexpensive reagents and equipment. We want to conduct the assay in relevant physiological conditions. We want it to be reporter-free, and we want to be able to test multiple proteases of the same family and maybe expand the assay out to other viral protease families without much modification of the assay. We want to provide good protocols and demonstrate robust performance with the ability to conduct high throughput screening. Hopefully the assay can produce similar results to the gold standard approach that people use, which is the live virus assay, And in an emergency situation, like a situation back in March, we want to be able to deploy the assay relatively quickly. So we set out to develop our assay, which is based on reversing protease-mediated cytotoxicity. We hypothesized that expressing 3CL proteases within mammalian cells could lead to toxicity. We're not the first person to try this. In fact, other groups have demonstrated this for other virus families. And we thought that protease expression could lead to cleavage of host proteins and downstream cytotoxicity. Of note, we haven't really delved into the mechanism by this. And we thought an assay of this nature would have specific advantages. One, it does not involve an exogenous reporter, just you need a cell. There's multiple methods to quantify cytotoxicity that already exist, and many labs have their favorite, and they're pretty good at churning through it quickly. And you only need expression constructs, which can be ordered from oligosynthesis companies. Finally, a lot of the reagents, such as cell lines, are widely distributed and available, and many labs already have experience working with them. So getting into the meat of our assay, when we expressed the SARS-CoV-2 3CL protease and 293 T cells, we saw quantifiable cytotoxicity. And we capture this data with crystal violet staining, which is relatively cheap and efficient. And so just showing you what this data looks like, when I express EYFP in cells, they grow pretty well, but when I express the SARS-CoV-2 3CL protease, I see a marked decrease in their growth. In fact, I can rescue that decrease by mutating the catalytic residue of the SARS-CoV-2 3CL protease. And I can easily elute the stain and quantify with a spectrophotometer, which is demonstrated on the right. Since we saw that we could reverse the cytotoxicity by genetically inhibiting the protease, we thought we could use a well-characterized 3CL protease inhibitor, GC376, and see if we could reverse cytotoxicity. And that is in fact what we did. And we saw that this resulted in quantifiable rescue of cytotoxicity. So next, we wanted to test not just against one compound, but see if we could capture others, and then also see if we could determine the potency at which these compounds act. So I'm gonna just walk you through the graph I'm gonna show, because I'm gonna show quite a few of them. On the y-axis side, the relative growth, which is normalized to essentially the vehicle-only condition. And then on the bottom, I have increasing concentration of the drug in log scale. So if I plot some data for GC376 against SARS-CoV-2 3CL protease, you see that there's a dose-dependent activity. I can fit a curve to it and calculate the EC50. I can also, at the same time, test the compound against EYFP expressing cells and determine the cell viability, which I like to plot on the same plot. Because our assay is dependent on cell growth, if the compound inhibits cell growth, obviously that can confound the assay. So it's a good thing to keep in mind. And then I can also calculate the toxic concentration, at what concentration the compound becomes 50% toxic. For GC376, it's not. So next, we turn to known SARS-CoV-2 inhibitors, and to see how well our assay mirrored results that were already published. The GC376, which I just showed you, another compound, compound 4, and saw dose-dependent activity, and then a third compound, compound 11A, which again, we observed dose-dependent toxicity. We wanted to match up with previous data regarding live virus assays to see how similar assays were. And so GC376, our EC50 of 3.3 micromolar matches up pretty well with literature reported values. You'll notice that there's quite a large range, and that may speak to inconsistencies with how people do the assay. Some like to quantify using cytopathological effect, and some like to quantify by qPCR or plaque assays, which can sometimes lead to different values. We looked at compound 4, again, we saw similarity. And then we looked at 11A, actually we saw about an order of magnitude difference between our assay and the literature reported value. So we actually went ahead and repeated this with the live virus assay and came up with a value that was much closer in line with what we found with our cell-based assay, which indicates that we are somewhat in the same ballpark as the live virus assay. We also looked at a few compounds that were reported to work in vitro that failed to work in a live virus assay, and just wanted to check in where our assay stood within that, whether it was telling us more about the in vitro assay or whether it was telling us more about what the live virus. And in fact, when we test these compounds, we don't see any activity, which suggests that our assay can differentiate between compounds that are active in vitro and compounds that are active against the live virus. Next, one of the big strengths of our approach, we think, is the ability to just test quickly a bunch of other 3CL protease enzymes to get a sense of how broadly active these compounds are. And we first just turned to a set of 6, which were some distantly related from SARS-CoV-2. We've actually expanded this out to about 17 enzymes currently, but I only focused on data for these 6 initially. So next we wanted to see the search space between the 3 compounds we tested and these 6 proteases, of which I've already showed you SARS-CoV-2. And this is a lot of data, so I'm just gonna focus in on a couple interesting ones. One, you'll notice for an avian coronavirus over here, we saw that compound 4 showed quite potent activity with 0.058 micromolar EC50, which is interesting and speaks to the strength of our approach, and that even though compound 4 was not designed as an inhibitor of this protease, it shows pretty strong activity. The other thing that was interesting was compound 11A, which does not show any activity against SARS 3CL Pro, which it does show pretty good activity against the SARS-CoV-2 3CL Pro. And then we saturated out the space, and just quickly, we found that GC376 and compound 4 seem to be broadly active against the 6 that we tested, although variable potency. We wanted to try and get an idea of why 11A showed this narrow activity and what kind of information we could draw from that might further guide drug development. So we looked at these 2 proteases via structure, and just for
Sam, your 2 minutes warning.
Okay. Okay, just you can see the compound may dock into this space. This is a zoomed in, and we wanted to look at structural differences between these structures. You'll notice that the S2 pocket is this boxed in region, which has quite a difference in structure, whereas MERS, it's quite closed off, and SARS-CoV-2, it's quite open. Looking at the differences between these 2 compounds, I wanted to highlight these 2 regions of the compound. While they're not the only different regions, you'll see that there's quite a different size and shape between GC376 and 11A. So we hypothesized that this may be playing a role. But because there are some other differences between GC376 and 11A, we synthesized a different compound SL4241, which is almost identical to GC376, except for this P2 substituent that docks into the S2 pocket of the 3CL protease. Interestingly, when we test this compound, we found that it was active against SARS-CoV-2, but not against MERS, which speaks to this larger, bulkier substituent that may prevent broad activity of these protease inhibitors. Next, we moved into high throughput drug screening just to see how well our assay performed at this scale. And I will move quickly through this because of the time constraints. And so we first optimized conditions and tested a small library of about 200 drugs that consisted of general protease inhibitors and then some with known structural similarity to 3CL proteases. And we spread this across 4 different plates. And on the y-axis, I'm going to show you the robust z-score, which is a measure of hit strength. And we identified 2 hits in addition to our controls, one of which was GRL 0496. We tested GRL 0496 in our assay and confirmed its potency. And then we saw if our assay could inform live virus testing. And so we tested this against the live virus, and we found that it was effective against the live virus. And finally, we looked at other viral protease families just briefly to see if our assay would be compatible with other families. We looked at a 3C protease, which is quite similar to the 3CL proteases. And again, we saw activity with known protease inhibitor rupintravir against this 3C protease. Next, we also saw that this was in line with reported values from live virus data. Next, we looked at the PLP from SARS-CoV, and we found that this inhibitor GRL 0617 showed dose-dependent activity against the PLP, which was promising as well. And this was in line with some of the live virus literature reported values. So to quickly summarize, 3CL proteases cause toxicity when expressed in 293 T cells. Incubation of protease expressing cells with protease inhibitors rescues this cytotoxicity. it's comparable. The output from our assay is comparable to output from live virus assays.
Sam, you're
It
quite a
allows-
bit over the time limit. That's
Okay.
good.
All right. Well, then I will wrap things up and wait for questions. I just want to say thank you.
Thank you.
Hey, Sam, I just have a really quick, simple question, and that is, are you guys planning to scale up and perhaps test some larger libraries beyond the 200, or the 180 component library that you originally tested?
We thought about this. I think at this current stage, we may hold off on that. But I think we're more interested in expanding out into some of the other 3CL proteases. But our lab is working on some other drug screening properties. And again, we're more interested in multiplexing the assay rather than kind of testing one and going out broadly into compound space.
Okay, cool, thanks.
Yeah, I mean, this is actually an important question because obviously the domain of protease inhibitors is not that huge, and so the biggest value of this would be in screening larger library of potential combinatorial compounds or other sort of large scale libraries of diversity compounds rather than the ones that are already known. So. But so one question is about the scalability of the approach in terms of even if you're not planning to do it in the lab, what do you think of the potential to scale it up?
Yeah, I think the assay is quite scalable. As to our initial next step, we're not quite sure whether we're going to truly scale it up.
Yeah, I understand.